Okay, so this is all about SARS CoV-2, which is the virus that causes COVID-19. And so your two I can statements are I can describe the unique characteristics of the SARS CoV-2 or COVID-19 virus. And so I just wanted to point out that this is the virus. It is an RNA virus. It is produced, so it has genetic material that is surrounded by a capsid, a protein-like shell. And within that protein-like shell, there are these protein spikes or clubs, you'll hear them called sometimes. Normally you hear spikes. And that is why it's called a coronavirus, or that's why it's in the coronavirus family. Corona means crown. And so these spikes kind of make this look, I guess, like a crown. Within these proteins, there is a receptor binding domain that binds to your ACE2 receptors, which are receptors you have on your cells. And so that is how it gains entry into your cell. It's also surrounded by an envelope that allows it to fuse with your cell membrane. So this would bind to an ACE2 receptor on your cell, and that would kind of help it dock, and then it would fuse with your cell and release this genetic material into your cell. There's a lot of different treatments that we're working on, trying to figure out. Science, not me personally. But one of the things we're doing, obviously, is we're working on getting vaccinated for this. We're working on a vaccine. There are actually almost 250 vaccines in development. There are 63 that are in like the clinical trial-ish stage, and there are 179, I think, as of making this video, that are in the like they're still just being developed in the lab. They're not being tested on anybody yet. But the one that seems to be making the most progression is the mRNA vaccine. And so let's just talk about that really quick, how that works. So as you remember, in your cells, you go through central dogma. You go through that gene expression by taking your DNA, transcribing it to RNA, and then translating it into protein if it's mRNA. And if it's tRNA or whatever, it goes whatever. If you put any mRNA in your cell, your body is going to say, oh, mRNA, let's translate it. And it's going to go to a ribosome, and the ribosome is going to make it into protein. So what we've done is we've used that idea because the code is universal. Remember that. So AUG will always be methionine, okay? So what we do is we inject mRNA with a lipid coating. That lipid coating is going to bind with your cell membrane. That mRNA is going to be basically through endocytosis. It's going to be sent into your cell. Let me move my head. And then your cell, not knowing any different or any better, is going to translate that using your ribosomes. This was another picture that was kind of good. So the mRNA comes in, it's translated through your through your ribosomes to make proteins. Those proteins are called spike proteins. And then your cell is going to put those on its surface. That's going to activate different parts of your immune system. Some of these proteins are also released. Those released proteins will be taken up. This should be two different cells. These proteins are going to be taken up by macrophages or other the immune cells, dendritic cells, macrophages, things like that. And they're going to put them on their surface with an MHC class 2, which is going to activate your T helper cells, which will then activate your B cells to make antibodies. Okay, so that's one kind of route. And the other is that your cells can post these on their surface using an MHC class 1, which will activate the cytotoxic or killer T cells. Those will also activate the immune system, but they will kill this cell because they think it's infected. And so either way, your immune system is getting activated in multiple ways, creating this huge response. The great thing is when your body is done with mRNA, you may remember this when we talked about gene regulation, it degrades it, it breaks it down. So when we're done with this mRNA, your body's like, we don't need it anymore. We're going to break that mRNA down. These proteins, they get broken down. So literally once this is over, once you've mounted this immune response, there's nothing of the vaccine left. It's all gone. The only thing you have left is that immunological memory. And because you're not inserting any part of the virus into your body, there's no chance of you catching the virus. Now, because there's this cell death going on, there's this activation of the helper T cells and the B cells, are you going to have some side effects? Yeah. You could have some soreness. You could have some fever, things like that. That's telling you that your immune system's working. Once that's over, though, all that's left is your immune memory. And that's it. So I hope that helps. If you have any questions, let me know.